This lecture is on gram-positive bacilli, or rods. We'll be discussing the aerobic gram-positive bacilli. The best way to learn this information is to group it into three categories. You want to look at the non-spore forming gram-positive rods, the non-spore forming branching gram-positive rods, and the spore forming non-branching gram-positive rods. This material is found in Chapter 16 of your textbook of Diagnostic Microbiology, 5th edition. So the non-spore forming gram-positive rods we're going to look at are Corynebacterium, or also called coroniforms or diphtheroids, Listeria, Erysipelathrix, Acronobacterium, Lactobacillus, and Gardnerella. Corynebacterium, we will look at diphtheria, Jacquium, Uroliticum, Pseudobacterium, Striatum, and Xeris. Corynebacterium is normal skin and mucosal flora. You'll also hear us call them diphtheroids or coroniforms, which mean club-shaped. They're gram-positive rods that are pleomorphic in shape, and they do not form spores. What they look like on a gram stain is a cylinder long tube with maybe a blip on the end that gives it that club appearance. You'll hear the term Chinese letters or palisade, where they line up on side to side when you talk about a gram stain of a Corynebacterium. You can see in this gram stain how these organisms stack on top of each other. Sometimes there'll be Chinese letters forming V's and L's and Y's. Corynebacterium are facultative anaerobes. They're small gamma colonies on blood agar. They have that Chinese letter appearance, which I spoke about earlier. They're catalase positive. That's an important characteristic. They're non-motile, and they're frequent skin contaminants. So let's discuss Corynebacterium diphtheria. That's a significant pathogen. It's one of the organisms that's in the childhood vaccinations now. Its virulence factor is the diphtheria toxin. So in strains that are infected with the bacteriophage, which carry the TOX gene, they produce an exotoxin which blocks protein synthesis. These destroy the host cell, and they may be absorbed into the heart and the nervous system and cause damage there. To give you an idea of how potent this toxin is, so it's lethal at 130 nanograms per kilogram of body weight, and that translates to, it would only take 3 ounces to kill a person that weighs 150 pounds. Corynebacterium diphtheria causes 2 forms of disease. The cutaneous form causes non-healing ulcers. The respiratory form is found worldwide, but it's not as common in the United States thanks to the vaccine. Diphtheria causes an upper respiratory infection. It's transmitted by droplet or hand to mouth. Bacteria multiply and release the toxin. It destroys the tissue. An inflammation response forms, and it causes this pseudomembrane, and the pseudomembrane eventually could lead to suffocation. And that pseudomembrane is a key characteristic of Corynebacterium. It's treated with an antitoxin, and it's prevented by the immunization, the diphtheria and tetanus combined. And you can see this formation of the pseudomembrane. So how do we diagnose Corynebacterium diphtheria in the lab? Well, it's a gram-positive rod, pleomorphic in shape. That means that there are different shapes and sizes present. They have a darker staining area of the cell. These are those metachromatic granules. And if you use methylene blue stain, you would actually see these. And the next slide shows the stain with methylene blue, these metachromatic granules known as Bates-Ernst granules. And what that is it's a pocket of inorganic phosphate or nutrient reserves. and you can see these in the picture next to it. On sheep blood, it's a slow grower. It's usually pinpointed at 24 hours. Serum or blood containing media. If you want to get it to grow a little better, you can use low-floor media, which enhances the development of the metachromatic granules. One of the agars you want to associate with the isolation of Corynebacterium diphtheria is Tinsdale agar. It's a selective agar. It has potassium tellurite, which inhibits non-coroniform bacteria. It's also a differential media in that the tellurite reduction produces a brown or a black colony, and cystinase causes a halo around the colonies. You got to be careful though, because Staph aureus can produce brown colonies also. So here's a picture of what the Corynebacterium diphtheria looks like on Tinsdale to the left, and then you also see Coroni on the chocolate tellurite agar. You want to associate Corynebacterium with ELK test. This would be an ASCP question. They love these kind of things. Immunodiffusion test for toxin production. It's a strip of filter paper that's saturated with the diphtheria antitoxin, and it's embedded in the agar media. So you take your isolate, and if you think you take your isolate, you take a positive and a negative control, and you're going to streak on agar parallel to each other and perpendicular to the paper. You'll incubate it for 1 to 2 days, and the lines of precipitate will form in the agar when the toxin interacts with the antitoxin, forming an arc of identity. So this is the way the ELK test works, and this is what the ELK test looks like. So the streak, the paper in the middle is soaked with the antitoxin, and then the streak at the very top is our positive control. And as you can see, coming off of the positive control, you have 2 lines that are forming a V there. And on the bottom, you have 2 lines that form a V. The one in the middle is our unknown, so we're trying to figure out if it's positive also, and you can see that it is. You have the 2 lines forming the V at the top and the 2 lines forming the V underneath. And then the negative control at the bottom, there are no lines on the top or the bottom forming a V. So Corynebacterium can be normal skin flora. Usually when we isolate them in blood cultures, we consider them a contaminant. You have to be careful though, because besides diphtheria, Jacquium is one of the ones that you'll see in catheters or prosthetic devices. You will associate it with valve endocarditis. Urolyticum is associated with a urinary pathogen. It's lipophilic, so it loves fats. It's a strict aerobe, and it's highly resistant. Other non-spore forming gram-positive rods. Urethia is actually a gram-positive cocci that appears like a rod. It's oral normal flora. It can cause endocarditis or wound infections. The other one is an undesignated CDC version of a Coroni group. That's where they just kind of group everything together, and they're opportunistic or nosocomial infections. So Listeria monocytogenes is a vaginal and intestinal normal flora in humans. Clinically, infection due to ingestion of contaminated food. It has virulence factors of hemolysin, which is Listeria lysin O, and this prevents phagocytosis. Listeriosis is sepsis or blood disease. It occurs in neonates with a fatality rate of greater than about 50%. It occurs in pregnant women and can cause spontaneous abortion and stillbirths. We see it in immunocompromised patients, and it also has a predilection for the central nervous system. Okay, so Listeria monocytogenes cultures, we usually use spinal fluid, blood, amniotic fluid. They're facultative anaerobes. They grow on routine media such as blood and chocolate. They prefer increased CO2, so therefore they're capnophilic. The colonies resemble Streptococcus agalactiae. You can also enrich growth at 4 degrees C, and that's called cold enrichment. Okay, so they're gram-positive rods. They're non-spore forming. They form small beta colonies on blood agar. Sometimes the hemolysis is hidden under the colony, so you'll have to move it aside to see it. This is a picture of the blood agar with the beta hemolytic colonies on it. Listeria monocytogenes are catalase positive, so that differentiates them from the Streptococcus. They're bilesculin positive, and they're motile at 25 degrees. Their motility is very unique. They have what's called tumbling motility at 25 degrees. So in a liquid broth, you inoculate a tube, you incubate it at 25 degrees, and one at 35 degrees, and then you observe it under a wet mount for this tumbling motility. And you'll see it positive in the broth at 25 degrees. And on this slide is a website on YouTube where you can go and see this. In semi-solid agar at 25 degrees, it has this umbrella pattern. And you want to associate the term tumbling motility and umbrella pattern motility with Listeria. If you look at the tube in the picture, right under the surface of the tube, you'll see this thing that looks like a little cap. Well, that's what they're calling the umbrella motility. Listeria monocytogenes is also positive CAMP, which distinguishes it from other Listeria species. But instead of being an arrowhead like Strep agalactiae, it is the bottom one on this picture that shows the blockhead positive CAMP. So here's a differentiation of Listeria based on catalase, bilesculin motility, beta hemolysis, and whether or not it'll grow in a salt broth. Erysipelathrix comes from a domestic swine as a major reservoir. It's an occupational hazard of butchers, vets, fishermen. It causes erysipeloid, which is a red skin lesion in animals and humans, and this can disseminate into endocarditis and bacteremia. Erysipelathrix is a gram-positive rod. They're catalase negative. They can be alpha or gamma hemolytic. They're non-motile, and they produce H2S on a TSI slant. They're cultures, usually from skin biopsies or blood cultures. They grow on routine media such as blood, chocolate, CNA, and PEA. They're very small colonies at 48 hours, so you have to be careful that you don't miss them. Acronobacterium, there are 9 species, only 3 of them are human pathogens. The most common Acronobacterium is Hemolyticum. This causes pharyngitis in teenagers. It's a facultative anaerobe. It's beta hemolytic, catalase negative. It's reverse CAMP positive, and it inhibits Staph aureus hemolysis. So this is a reverse CAMP test. So what you have here is you have your streak of Staph aureus, and then when you do the Achromobacterium hemolyticum to the staph perpendicular, you get a reverse arrow instead of like you do with the group B on the right. Lactobacillus is normal vaginal flora. It produces lactic acid. It reduces the vaginal pH and inhibits growth of other organisms. It rarely causes disease. Probiotics are really huge out now, and Lactobacillus acidophilus is the bacteria that they use in probiotics. It's a non-spore forming gram-positive rod. They're medium to long rods. They're aerotolerant anaerobes. They're catalase negative, and they're non-motile. They're tiny alpha colonies on blood. Gardnerella vaginalis, you want to associate with vaginal infections. They're pleomorphic coccobacilli. The cell wall has gram-positive characteristics, but sometimes they'll stain gram-variable. They're non-modal, they're facultative anaerobes. They're normal flora in about 50 to 70% of the women, and they're linked to bacterial vaginosa. So any questions concerning bacterial vaginosa, you want to look and suspect maybe Gardnerella. What happens in bacterial vaginosis is there's a change in the normal flora. So there's a decrease in the number of lactobacillus and an increase in pH, and this allows Gardnerella to grow. You end up with a foul-smelling discharge like an amine odor. You'll do a sniff test on wet preps. When patients come into the ER, they'll collect a saline tube with a swab from the vaginal area. We'll take a drop of that and add 10% KOH, and you smell like a fishy odor or an amine odor, and that's positive. Untreated can lead to pelvic inflammatory disease and urinary tract infection. Culture is not recommended. Bacterial vaginosa, Gardnerella is hard to grow. Usually what they'll do is this wet prep, and they'll look, or gram stain, and they look for these clue cells. And what clue cells are squamous epithelial cells with bacteria that are clustered at the edges. Okay, so this is a picture of clue cell on a wet mount. This is squamous epithelial that's full of bacteria, and the bacteria mainly conglomerates around the edges, and it kind of gives it a furry looking appearance. And actually on the wet mount you can see the Brownian movement, and it makes it look like the bacteria is moving even though Gardnerella is non-modal. The only time we do cultures if it's an extravaginal site. It will grow on chocolate blood, but it does not grow on MacConkey. It's a slow grower. We can do a presumptive identification if we gram stain it and we see these small gram-variable coccobacilli, if catalase and oxidase are negative, and if you do a hipparate hydrolysis and it's positive, you can call it presumptive Gardnerella. Okay, so now let's talk about the non-spore-forming branching gram-positive rods. These are what we call our aerobic actinomycetes. We're going to talk about Nocardia species and Trophirma species here. A couple of the others that are important in the clinical laboratory would be the Streptomyces and the Rhodococcus. So Nocardia species are found in the soil. There's about 88 species. 46 are implicated in human infections. You usually see these in your immunocompromised patient, and you'll see them as a pulmonary infection or cutaneous. 10% of some Nocardias occur though in healthy patients, and 40% of Nocardia is diagnosed at autopsy. So in Nocardia species, you have two disease processes. The pulmonary, you inhale the organism, so you get a pneumonia, which forms into an abscess, which causes tissue necrosis. It may disseminate. The cutaneous is called actinomycotic mycotomas. Nocardia brasiliensis is one of the organisms that cause this. And one of the key characteristics of this is the draining pus with sulfur granules. Nocardia species are aerobic gram-positive rods. They're morphologically similar to fungus, but they are true bacteria. They form beaded branching filamentous on a gram stain, and they're weakly acid fast. And this is what it looks like on a gram stain. You can see on the gram stain on the right the beaded rod, and on the left is actually a fungal stain showing gram-positive branching. They grow slow. It usually takes a week or longer. They're waxy, chalky, crumbly colonies. This is one of those ASCP-isms. They'll call it a crumbly colony as a description, and that wants to clue you in as to you're looking at possibly a Nocardia. Here's it on blood agar, and you can see that it, when you touch this colony, it would just fall apart. They're filamentous branching organisms. They're weakly acid fast, so you want to associate Nocardia with a weakly acid fast. You can use a Ziehl-Neelsen, which uses a heat, or you can use a Kenyon. So when you talk about an acid fast stain, we kind of mentioned this in earlier lectures. Your primary stain, once again, is carbofuchsin. Then you're going to decolorize it with an acid. Then you're going to counterstain it with a methylene blue. And so reddish purple filaments would indicate partially acid fast, and blue would be negative. And here is a gram stain compared to a partially acid fast. In the gram stain, they stain gram positive, which makes them purple, and in the acid fast, they're going to be pink. Trophirima whippelia is Whipple's disease. You want to associate it with that. That's an intracellular pathogen. It's uniformly fatal if untreated. You have diarrhea, weight loss, malabsorption, and you have to identify this by PCR or 16S rRNA. Streptomyces you'll see sometimes in the clinical laboratory, as well as the rhodococcus. So now let's go to the final category, is your spore-forming non-branching gram-positive rods, and this is just your bacillus. So your bacillus is a spore-forming non-branching bacilli. Your spores they're protective. They're metabolically inactive. It's kind of like a survival stage. They're resistant to biocids, and they have a thicker outer wall. They're found in nature. They can be lab contaminants. Bacillus anthracis and Bacillus cereus are the two most important pathogens, so it's important that you rule those two out. It's a large aerobic gram-positive rod. Spores, they don't stain by gram stain. They appear as holes. Beta hemolytic, except for Bacillus anthracis, and they're catalase positive. So here's a gram stain, and you can see where those spores are in the hole. They're huge. You won't see anything bigger than this. They're catalase positive. They have aerobic spore formation. They grow on blood. They grow on PEA, but they will not grow on enteric agars like MacConkey. So Bacillus anthracis causes anthrax. It's usually in herbivores. It is also a bioterrorism agent, so it's a notifiable to CDC. It has virulence factors, which consist of a glutamic acid capsule and exotoxins. So here's Bacillus anthracis. You got a clear zone on the left. You can see it as a capsule. It's one of the virulence factors. And then the diagram on the right shows you how the protective antigen, the lethal factor, and or edema factor affect how anthrax gets into the cell. The protective antigen actually just allows it to adhere to the cell surface, and then the lethal factor and the edema factor work with the protective antigen and can cause cell death. There are three forms. There's cutaneous, pulmonary, and gastrointestinal. So cutaneous anthrax starts out as a pimple, vesicles, and then it goes to erythematous ring. It produces necrotic lesions which turn black. Inhalation of anthrax causes a pulmonary disease. Wool sorter's disease is what this is called. It's a flu-like phase followed by respiratory distress, coma, and then eventually death. GI anthrax is ingestion, and you get pain, nausea, and vomiting. So on the gram stain, you got very long chains. Sometimes you'll hear them referred to as bamboo shoots, and they're square ended. They're non-motile, non-hemolytic, Bacillus anthracis is. You'll have a large flat colony with irregular edges, and this is, they call it the Medusa head colony. That's one of those ASCP-isms. So Bacillus anthracis is an aerobic or facultative anaerobe. It's a spore-forming gram-positive rod. It's non-beta hemolytic on blood agar. It's non-motile, and it's catalase positive. If you have an organism that you suspect is Bacillus anthracis, you want to definitely work it under a safety hood. And if you can't rule out that it's not Bacillus anthracis, then you must send it to your state laboratory or CDC for confirmation. Bacillus cereus is beta hemolytic and motile, so that distinguishes it from Bacillus anthracis. Cereus produces an enterotoxin, which causes food poisoning, and it's usually self-limiting, meaning that it will clear on its own. Here is the colony morphology for Bacillus cereus. It looks like a frosted glass colony, and it is beta hemolytic. And this is a flow chart on how to identify these gram-positive rods.